In this video, I'm going to show you how you can complete the Simulating Segregation Quick Lab. Let's get into it. All right, so let's see how we can go ahead and complete this quick lab. It seems complicated, but really once you figure out what's really going on here, it's really not that complicated. I think the hardest part is probably determining what you're going to use as your alleles. But first things first, let's go to our actual assignment here, the quick lab, simulating segregation, worth a big 40 points. All right, so this is what it looks like when you first come in here. And if you click Start Attempt, the first thing you're going to see is all of the instructions. And there seems like a lot of words there. But if you read through all of this information, you will find that it gives you very clear step-by-step instructions. So, I mean, you can kind of skip through the guiding questions and the objectives, although those are important to keep in the back of your mind as well. But the assignment itself tells you exactly how you're going to do this assignment. So the first thing you need is 40 of something. Whatever that is, you decide whatever works best for you. The easiest thing would be to use, if you have 40 marbles, that you have 20 of the same kind of two different kinds of marbles. Or let's say that you have dried beans in your house. You got some red beans, you got some black beans, or you got some white beans. Any two colors would be fine, just so you can kind of differentiate between the two things. In my case, I have pennies. So I'm going to take, I have 40 pennies here, and 20 of them I'm going to mark with a marker on the head side so that I know that that's that kind of allele. So that's going to be my dominant allele. 3. I need 10 of them. 4. No, I don't. I need 20 of them. 5. And I'll speed this up for you. 6. 8, 9, 10. Okay. I don't need the marker anymore. All right, so I need 10 unmarked. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 on this side, and there should be 10 on this side. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 on this side. All right, now I'm going to cover them. I'm going to shake them up, because remember that alleles are randomly assorted and randomly selected. All right, so there we have our alleles. So we have one parent, and they have 10 chromosomes, and they have 2 alleles for each chromosome. So there's 20 alleles in here. All right, there's half of them are dominant and half of them are recessive. Okay, this parent also 20 chromosomes, 20 alleles. And what we're going to do is we're going to randomly select 2 alleles, one from each parent, and we're going to use that on the next page of our assignment. So let's go back to here and click on this, and you can see that we've got some instructions there. All right, so we're going to call the pennies that are marked with black, they're going to be our big B. And then the ones that don't have a black mark are going to be our little b. Okay, you can use any letter you want, but you use a capital case and a small case of whatever letter you choose, but you only use one letter. Okay, our 2 letters are going to go in the genotype, and our actual allele is going to go in the phenotype. So when we get down here to our answer box, we can either use the table tool and create our own table, or we can just copy this one because it's already kind of set up for us. So I'm just going to highlight all of that. I'm going to click and drag all the way down from the top to the bottom. Then I'm going to copy. Then And I'm going to click in the box and I'm going to do paste. Voila. So I don't have to do anything in the trial number. It's already numbered for me. But here go my results. Okay. So now that we have all of our alleles in our 2 different cups from our 2 different parents, we can start doing our trials. So to do that, I'm just going to randomly shake them up a little bit. I'm going to pull 1 penny out of each jar, put them heads up, and I have two pennies that are marked black. So that's my dominant allele. So I'm going to put a big B and a big B under genotype. Okay, my phenotype is going to be homozygous. Dominant. That's homozygous dominant. So now I'm going to do my second trial. So I'm going to put these back. I'm going to shake them up. Because remember, the allele that the offspring receives is completely random, right? 50-50 chance of getting either the dominant or the recessive. So I'm going to pull 1 out of each, put them both heads up, and I have 1 black mark, 1 without. So I'm going to go B, big B, little b. Okay, that would be heterozygous. Or you could say heterozygous. And I guess you should also mention that this would be black. Okay, this one would also be black. So I can just copy and paste that. All right, let's do one more, and then I think you get the point. You do this 20 times. Here's my third trial. And I'm going to put them heads up, and I get another one, another BB. So I can copy that. Whoops, I cut it. Let's paste it back in. Paste, paste. Copy that. Paste that in. And you just keep going right down. We'll do one more just to see if we can get a homozygous recessive. Let's see if we can. Whoops, I looked. That's cheating. Okay, let's see. Oh, I did it. Homozygous recessive. So little b. And we're going to go with homozygous recessive. I know I'm copying this and it's not the correct thing, so I'm going to have to change it, but at least I don't have to type homozygous again. Recessive. And that's going to be not black. Our recessive trait. Okay? And you're just going to keep going through, and you're going to do that 20 times. Okay? Now, once you've done that 20 times, you'll go to the next question. And it's going to ask you to revise this investigation to simulate the crossing of 2 genes. In this case, we only crossed 1 gene, right? And we picked 1 allele for the gene from each parent. What if you were trying to cross 2 genes? How would you change this simulation to simulate crossing 2 genes? The rest is up to you. Thank you so much.